Good day, everyone. My name is Ken Buetow. I'm from Arizona State University, and I'm the Director of Computation Science and Informatics of the Complex Adaptive Systems Program and a professor in the School of Life Sciences at ASU. In today's session, in this specific session, I'm going to be talking about understanding the genetics of common complex disease with a focus on using big data approaches to see the emergent whole. As this audience is aware, we are in the midst of a transformation of the biomedical universe, one that encompasses and captures the concept of personalized medicine. Personalized medicine being delivering to the right individual the right care at the right time and in the right place. This transformation of medicine is both a revolution, in particular in the form of translation to precision medicine using one's molecular constitution and the molecular constitution of your disease to drive your therapeutic intervention. But it's also simultaneously a renaissance, a return back to the days of medicine and medical delivery where we not only delivered disease, not only treated disease, but also treated the individual as a whole, the context in which they lived, the exposures of which they had, the preferences for their particular care patterns. We, in this war on disease and in personalized medicine, have very many exciting developments. This is a cover from Time magazine in 2001 talking and heralding the promise of personalized medicine. In particular, this was focusing on the targeted therapeutic Gleevec and the suggestion that we may actually have a transformation in how we were going about treating cancer and the response to cancer to therapeutic interventions based on the use of molecular information. Shown on the next slide, though, is what is really materialized as we move to these targeted precision medicine interventions. While unequivocally, they do have components that are nearly revolutionary. We can see that individuals treated with one of the targeted therapeutics here have a long period of response that's basically was unheard of and actually looked transformational. Basically, in treating these diseases, these individuals in these portions of the curve basically had their disease disappear for a period of time. What's been disturbing or frustrating for us, though, is that what we've seen that even though we have these miraculous, nearly transformational periods, what happens is at the end of the day, though, almost everyone still relapses, and the disease, we don't get what's a durable response. And there's concern that this may actually exist not only in cancer, but many of the targeted therapeutics. But I'm going to spend my time talking about cancer in this instance as to why this may be occurring. And while we have tremendous insights that are coming from this new personalized precision medicine revolution, that in fact the problem is a little more complicated, and hopefully understanding how we might approach it so that we can change those temporary responses into permanent responses. So let me spend a minute or two talking about cancer, which is one of the better characterized common complex diseases at a molecular level, and share with you why perhaps these focus on a single entity and treating on a single basis is not necessarily what's driving the disease. The other thing I guess I'd encourage everyone as we start to move through this that I should have said up front is as we move through this, if you have questions or suggestions or want to know follow up, but for the question period, please enter those in as we go, and I'll answer them at the end of the talk. So as we walk through, cancer is, while we can look and focus on particular entities, we can see cancer, like many common diseases, is complicated. In fact, cancer is a complex adaptive system, and I'll walk through what that means both cartoonishly as well as in summary. So cancer, we know, emerges by the accumulation of mutations from a largely normal base state to ultimately create or have hundreds to conceivably thousands of mutations that result not only in the generation of a different malignant state, but actually the development of a different organ, almost the development of organogenesis or evolution in action, where you not only have different changes in the molecular state of cells, but the cells themselves in the phenotype actually, what one sees is actually one creates a whole new population of cells that may or may not be the same as the rest of the surrounding environment. So you have hundreds of alterations that generate this complex state. We see that, in fact, we can generate these changes by mutations, somatic mutations or germline mutations, but nucleic acid mutations aren't the only way this can occur. It can also occur by changes in copy number, so more than just point mutations. It can occur due to changes in gene expression level, due to changes in epigenetic modifications. They're not occurring necessarily at the nucleic acid level specifically, not specific nucleic acid modifications. We can see that there are extra gene events through microRNAs and the rest of the transcriptome that generates alterations, as well as processes, post-translational modifications to the genes and the proteins that can make dramatic changes to the specific state of genes and the phenotype independent of the nucleic acids themselves. We also know that there are a number of processes, and for more than a decade, our colleague Weinberg and Hanahan demonstrated, at least in cancer, and we know in other common diseases, common processes that get modified. Shown here in cancer, as we can see things like growth factor independence, evading apoptosis, the telomere maintenance are fundamental processes that need to get changed. But what one drills down a little bit more, one realizes that while one can simply state growth factor independence or evading apoptosis, these themselves are actually complex networks of multiple interacting genes that combine to produce phenotypes. And that these hundreds of mutations that occur can occur in different points in each of these different processes. So the hundreds of mutations in one individual can occur in one set of genes. In another individual, they occur in another set of genes in these processes, and yet in another set of genes in a different collection of network nodes in the specific processes associated with any given disease. We also know that the somatic basis of cancer isn't the only thing driving these phenotypes. One's genetic constitution can also drive these processes. And it's clearly emerging now that one's inherited variation can drive things such as your basic angiogenic processes, your cellular matrix formulation, and certainly in cancer and in many other diseases of interest right now, your immune response, whether you identify the disease and how you tolerate the disease as it's moving, as it progresses. But these not only have to do with internal processes that drive these, but the genetic constitution drives how you respond to chemical exposures, viruses, hormones, nutrition, and the whole onslaught of different types of factors that would be outside of your environment. So I think it's fair to say that cancer, it's not only complicated, it's very complicated. And it's through this complication that these complications, the evolutions, the changes in pattern and other, that are resulting in the ultimate failure and the complexity in designing these therapeutic interventions in common disease. In fact, this complexity has recently been embraced by one of the founders of the reductionist paradigms in cancer research, Bob Weinberg, who recently in Cell wrote that while we had hoped that looking at just single genes or single processes would give us clarity into the future and to a clear path as to how we would do our interventions or prevention strategies, he now actually writes that, in fact, we've got to come back and embrace this complexity if we truly want to understand common disease in general and cancer in specific. I metaphorically refer to this as where we stand today in the molecular biologic precision medicine context is we know a lot about the individual pieces. Our good 30 years of solid reductionist experiments have helped us identify the parts list. And we have now a rich lawn of this, but the picture they describe is actually quite hard to elucidate. And what we recognize is if we really want to understand the picture, we have to understand how all these pieces come together, the system by which they're assembled, and how the individual pieces interact. Sort of as the pointillists early on in the post-impressionist fashion learned that you could create complex images by combining the point colors with your brushstrokes, and by doing that, a picture emerges by considering those interactions and looking at the combination of the individual points. Suddenly, coherence begins to emerge, and not only coherence, we start to get a much more complex picture that shows not only how those points describe a specific individual piece, but they also are part of a much larger picture and puzzle, and we can see that where we may have even been focused doesn't necessarily describe the main center of action, which in this instance is more here, and while our attention was focused at the original points over here. So it's critical that we assemble these parts so that we can see the whole. So why is that the case, and why is embracing this complexity important? It's because the whole in biologic systems is more than the sum of the individual parts. If there's a short redefinition of what a complex adaptive system is, and a take-home lesson is the whole is more than the sum of the parts. So this complexity results and generates evolution over time, where the system isn't the same as it was tomorrow, as it was today, and as it was yesterday. It has adaptive behaviors. It responds to things that we do, both intentionally in interventions and our broader lifestyle and other components, why we need to embrace the other dimension of personalized medicine. It displays emergent properties, just as the pointillist, by putting primary colors together, could get a rich palette of colors without having to mix them on their palette. We can see that there are emergent properties that result in unintended consequences, and clearly require us to have transdisciplinary approaches to these problems, embracing the entire ecosystem, with embracing the interdependencies, and in particular, the information component thereof. So we talk about precision medicine as driving this, and clearly precision medicine is one of the big data components associated with doing personalized medicine, but we also realize that we need to be worrying about the clinical phenotype of individuals as well as their exposure. So we generally have a multidimensional big data problem. The genome's measured in gigabytes themselves, images or the phenotypes are distributed all over and are large and diverse, and other means of clinical characterization, such as imaging, provide as much or more data than the precision medicine molecular definition of the problems, as well as new generations of personal monitoring devices. In the news this year was Google's announcement of tools that will allow individuals to monitor in real time, all the time, their glucose levels, as well as a whole slew of personal monitoring devices that are going to be as large and conceivably dwarf what we see in the context of genomics medicine in terms of the data that they need to address, and that needs to be part of this holistic consideration of common disease. So while we can do interesting and important things today, what I want to focus on for the rest of the talk here is what does the future look like, and what are we and others trying to do to embrace this next big data future to address common disease. So where is it that we stand today, and where are we going with respect to the future of using all of this data to help us understand, intervene with, both in terms of prevention and treatment of common disease? And I guess following the old movie metaphor of The Graduate, just the same as he was given one word of advice as to the direction to go. For those of us in big data science and next generation big activities, we about one word to say: Google. And Google represents a model as to how we can go about embracing and understanding this data. But let me share, at least metaphorically, why I would invoke Google. Most of us are familiar when we do a Google search that as quickly as we start to type in a particular word that we're interested in searching, Google has already presents us with options of how, or anticipating what we may want to do based on detailed examination of integrated patterns and the emergent properties associated with the connection of individual datums from, millions to billions of different queries. So in our universe, what we want to be doing is mirroring what Google does. We want to actually know the interrelationship between all these components, and we have an opportunity to use the same class of tooling and analytics that Google has used to process these billions and trillions of observations to find the specific patterns that then we can reduce back to show both biologists and clinicians interesting and important patterns that exist in the data. Let me use another metaphor. Most of us are familiar with Amazon.com. And again, Amazon does a similar class of analytics by analyzing and taking together all of the books it has in its inventory. It looks to understand how all of those are interrelated, and looks for the underlying correlation and networks that brings them together, so that when I enter a keyword or look for a particular book, it can then examine the network of interacted or interconnected books, look for the nearest neighbors, and suggest to me other books that may be of interest to me. So what we want to be doing in biomedicine and in the search of common disease is to assemble this data in a concerted way, look at its understanding and understand it by joining and identifying the interrelationships and the interactions, and then not ignore them as we would do in a reductionist sense, but actually use those interactions to help us better understand the patterns and how we would both understand, intervene, and prevent disease. And this is all part of an emerging new paradigm of science. The fourth paradigm of science is a book written by Jim Gray from Microsoft about a decade ago now at this point, that talks about how we will need to be bringing into the biomedical space and other spaces the capacity to not only look at and generate data generating science, which dominates most of what we do in biomedicine today, and create data-driven science. And actually, I've taken the liberty of modifying even what Gray said. He used to describe this as hypothesis-generating science versus hypothesis test versus data science. I actually believe that data science can also do hypothesis testing, and I think the cleaner distinction is between data generating and data-driven science. And the reason is I look at the models such as astronomy and physics and economics, and we can actually test very rigorous hypothesis, for instance, in astronomy, without ever actually formally conducting the experiment where we, for instance, remove the sun from the center of our solar system and say what would happen if that occurred. Through data science, we can actually, from our observation basis, actually conduct virtual experiments and have the same power. And I think most of us probably would not contest that the law would not actually suggest that the laws of planetary motion are just suggestions or just hypothesis, but in fact are actually laws established by appropriate observation and the appropriate application of data science. So I'd like to, at least to this audience, pitch that we think that data science, as does Harvard Business Review, think that this is a critically important new direction that needs to be added into the portfolio of scientific investigations. And it's really predicated on the observation that at the end of the day, data is really powerful. And part of the promissory note of large-scale projects is that by having all of these data available, there's really cool things that we can do with these. And in fact, this is starting to begin to be introduced into biomedicine in general, healthcare in specific, in addressing what I like is the described in this instance is the Wanamaker dilemma, but helping us in particular in using and applying data science in our biomedical problems to address the class of problem that that Wanamaker almost a century ago cared about. And so if we talk about generating interventions based on molecular profiles and precision medicine, Wanamaker was very interested in knowing how to direct his advertising money. And what he was concerned about is that he didn't know where or who was responding to his advertising, and wanted to use analytics from observations to be able to help predict this, and was actually one of the pioneers of statistics and evidence-based advertising. Well, as you all are probably aware, this is now exploded. In fact, there are now very large-scale companies. In fact, if we summarize the market capitalization of the companies I have on this slide, whose whole business model is using analytics to predict who will do what when, as well as to drive people to particular components, we can see that this probably exceeds the amount of money that the pharmaceutical industry is spending in its research. So there's a rich quantitative base and arguably a significant proof of concept that with the right collections of observations, we have the analytic methods that would suffice to actually make the types of important observations, both identify the basic underlying etiologies, but then based on using this data science to make the appropriate predictions about who and how people will respond to particular interventions. Part of our challenge, though, of course, back to the sexiness of data science, is that we've got to transform our current paradigms. And I love, actually, this quote from Jeff Hammerbacher, who is, as you can see, in 2006 at 23, quite a scary age for me, given my substantive flock collection of white hair, is one of the first 100 employees of Facebook, and was charged with uncovering why Facebook took off in some universities and flopped in others. And he's currently now assistant professor in genetics and genomic studies at Mount Sinai. But part of his point was that it's really important that we start translating what's happening in this commercial sector into areas that in fact can both identify the basis of disease as well as help us predict who and how people will respond to our interventions. I think his more colorful presentation that it sucks that we're actually not doing this yet is a fair admonishment for us in the biomedical community. So we at ASU are actually attempting to get into this game, and I'm going to spend a brief vignette now talking about the basic capability that we're creating, and then I'm going to spend a little bit of time talking about how we're starting to apply this to common disease to hopefully realize the promise of big data in finding these emergent properties. So part of what we're doing at Arizona State University is creating a next generation data science instrument called, in this instance, the next generation cyber capability. So what do I mean by that? Well, we're quite familiar with large-scale data-generating scientific instruments. Shown on this slide are like the Mars rover or the Large Hadron Collider, and we know things like the Hubble telescope. Our intention at ASU is to try to create, for data scientists, a similar class of instrument, an instrument that allows us to do the powerful things that having the Large Hadron Collider or the Mars rover or the Hubble telescope enables other scientists to do. In this instance, we see this data science instrument is an elemental whole. And when I mean elemental whole, I mean that in the true physics sense, that an element is the smallest entity in nature that retains its essential properties. You can break it down smaller into its neutrons, protons, and electrons, but it no longer retains its elemental whole. And we see a data science instrument is composed of three essential components: physical capacity, ultra-high bandwidth networks, large-scale storage, and multiple flavors of computation. At ASU, we're connected to Internet 2 with 100 gigabit network connectivity. We have more than 2 terabytes of more than 2 petabytes of large-scale storage. We have utility computing, traditional high-performance computing as represented in a 1,000-node cluster, as well as a new big data computing where we have a 40-node Hadoop-class cluster that each of the nodes has 16 processors with 2 terabytes of memory and 250 terabytes of storage. But the physical capability is not enough. It needs logical capabilities, in particular different flavors of core software. I mentioned Hadoop and some of these other new big data capabilities, but also the traditional analytic pipelines that are part of the genomics array. It needs the management of metadata, data about data, as well as the semantics of how all these different pieces interconnect. And then lastly, and I would actually argue if there is a more important piece, it's complemented by a team, a transdisciplinary team that knows not only how to make the hardware and software work at first principles, but has domain knowledge that allows one to use this instrument, just the same as you have the JPL scientists that know how to run the Hubble telescope. It's a team of scientists that can funnel to the primary domain-based investigators the core findings and the core use of this instrument that supports their ongoing work. So here's a different cartoon that shows it all sort of brought together. So at the bottom of the slide here, you can see the physical capacity components that I just mentioned a moment ago. You can see the software components here that are demonstrated in this instance, that allow you to do particular operations, the analytic tools, the metadata resources that are part of this, and then completely on the right side of this slide, then a rich collection of different types of information and infrastructure that would support creating and driving this big data universe. So in this instance, our platform, we believe, then facilitates the conduct of data science experiments. So what we're doing now at ASU is turning this instrument, just the same as for the Hubble telescope, you pointed at a particular point in the sky. We're pointing this instrument at a particular point in the sky for us, the problem of diabesity, a true common complex disease. And what we're attempting to do is use this data science approach to try to find novel insights with respect to this particular disease and or collections of diseases. So I'm sure this audience is aware that there is a worldwide explosion, an epidemic of obesity and diabetes. Shown on this slide is a cartoon from our French colleagues that show the perhaps not surprising explosion of diabetes in North America and in Central Europe, and in the industrialized European countries. But what's also interesting on this slide is the observation that diabetes and obesity isn't only emerging in those universes or in those communities, but is also emerging in areas such as India, occurring in areas such as the Middle Eastern Peninsula. So a variety, and in other developing nations as well as developed nations. And actually this slide actually underestimates, for instance, the explosion of obesity and diabetes in China because it's not uniformly distributed. But if you go into the major urban areas in China, the rates of obesity and diabetes are as high as they are in North America, in Europe, and in the other developing nations. Interestingly, obesity has many manifestations, and problematically. So being obese results in many outcomes. You can get cardiovascular disease. I've already mentioned type 2 diabetes in the Diabesity framework. But one also can get liver disease in specific and cancer in general. Much of our focus in studying obesity has found that this is a product of very complex collections of interaction. And shown here is the 4-site map generated by the SIFT group that actually is almost purposefully unreadable in the sense that it describes from their examination the complex etiologic framework that's responsible for the development of obesity and its related phenotypes. Interestingly, in biomedicine, we largely focus on just that first central core here about physical activity, energy maintenance through metabolic and biologic processes. But as we can see, that core, while important, is embedded in a much larger network of interacting components. And in fact, those other interacting components are as diverse as food production, food consumption, social psychology, individual psychology, complex notions of built environment and how they drive physical activity, as well as then our more traditional biomedical focus on physiology. So while it's important to recognize the biologic aspect, the core individual biologic aspect, it's also important if we're going to embrace obesity and its outcomes, its origins and outcomes, that we know that that's embedded in a much deeper context. And we build, as data scientists, the systems that allow us to not only collect the molecular phenotypes, as we started with cancer, but also the psychosocial and other environmental factors as well. Well, I'm going to drill down a little bit, since we can't completely boil the ocean, and focus our attention a little bit on one of the dimensions of obesity and its morbid outcomes, and that's just citing a paper that came out, I think, about a week ago in The Lancet, that has re-emphasized that obesity is directly related to many cancer phenotypes. And shown on this slide is the top 5 cancers that are associated with BMI and weight, some of them actually in an almost perfect dose-response relationship. But we can see here that uterine cancer, gallbladder cancer, kidney cancer, liver cancer, and colon cancer all have highly significant associations with patterns of weight and with obesity. In the remainder of the talk, or for the next section, I'm going to focus a bit on one of the subdimensions of this and talk about both about liver cancer, and I'll share with you in a moment why I'm going to focus on that subset of the disease as we move forward. So why liver cancer, or the most common technical definition of a specific form of liver cancer, primary liver cancer or hepatocellular carcinoma. And I'm going to posit to you that liver cancer, given obesity trends, could be the lung cancer of the 21st century. And I state that in the sense with the evidence base that follows. But when I say the lung cancer of the 21st century, those of you who are familiar with epidemiology and the history of epidemiology is 100 years ago, lung cancer was a relatively rare disease. And in fact, on some of my mentors, my original clinical colleague mentors say that when they were training, it was considered to be an unusual cancer. But with the advent of smoking starting at World War I, but exploding around World War II, lung cancer suddenly moved from not only being a rare cancer, but dominating cancer, and the most common cancer and most common public health problem cancer in the developed and the developing and the broader worldwide exposure universe because of the explosion of its exposure, because of the explosion of smoking as its underlying etiologic risk factor. Well, I would argue that we've now established that there is a explosion in obesity. We've now established that liver cancer is associated with obesity. But one of the interesting trends associated with liver cancer is if we look at trends in cancer, and this is work published by my colleague Dr. McGlynn about 2 or 3 years ago, it shows that liver cancer, unlike most other cancers, is rapidly increasing in incidence. If you look at the cancer incidence trends for most cancers, they are either flat or declining, with 2 exceptions, liver cancer and pancreatic cancer. And liver cancer, as you can see over the last 2 decades, has essentially doubled in its incidence rate, which tracks, you'll see, with a changing risk profile for liver cancer. So historically, 20 years ago, liver cancer was associated with alcohol exposure and chronic virus infection. But in 2014, the major cause of liver cancer is obesity and diabetes. So we may be at the early end of a trend whereby we're going to see an explosion of liver cancer, not unlike the rapid increase of lung cancer that emerged with the chronic exposure to cigarettes and cigarette smoking. So let me walk through a little bit about liver cancer for those of you who are not liver cancer aficionados. Liver cancer starts in development of liver cancer with a normal liver, normally proceeds through some flavor of acute hepatitis that moves to fibrosis, cirrhosis, and then ultimately cancer. And what we can see is there's now, not only do we have the traditional risk factors of virus and alcohol and some rare environmental exposures, we have diabetes and obesity as primary drivers. So my group has been very interested in pointing our data science instruments at actually seeing if we can find some of the underlying components, what's driving or differentiating the people who go on to develop liver cancer, people who develop liver cancer versus people who don't develop liver cancer. And we've been doing that by using one of the molecular components in the precision medicine, inherited DNA variation. Partially because it's one of the, while it has the ability to change over time in somatic variation, it's one of the few invariant measures we can start with that we know has at least been present since birth for an individual and gives us at least a solid footing on which to start. In these, what we've been doing is using a mapping paradigm. There's lots of ways that we can map phenotype to underlying process or to molecular phenotype, family studies, gene-based signatures, which have a lot of cachet in the common molecular literature today. Our group has been focusing on a specific subset of these activities, and that's looking at network interaction signatures. Because again, reminding you all, I'm interested in the emergent properties. What happens when you combine components? So while we will look and we do characterize the individual gene-based components, we also want to look at what happens when you combine the genes in order to produce particular phenotypes. Our particular collection of tools has been looking at network analysis of constitutional variation using tools developed by a postdoc at the time, but now a faculty member at Northwestern University, Rosemary Braun, who developed a collection of novel tools for looking at the interactions of constitutional variation in producing phenotypes, called the Pathways of Distinction class analysis. And I point you, if you're interested, to this Pilos genetics paper that describes this in detail. I won't go into it today, other than saying it has the capacity to study the specific interactions between genetic variation and look at that relationship in those interactions to the outgoing phenotype of which one's interested in processing. So we applied this pathway of distinction analysis to primary liver cancer using a case control population, trying to find the underlying molecular mechanisms. And what I point to you in this, and the reason this becomes then big data, is we're looking at almost 1 million SNPs and variants as well as copy number probes in a population that measures in the thousands. And in this, and I realize this is a largely unreadable slide, but you can all look at it later, we identify a variety of very highly significant molecular processes that are driving the individual phenotypes. So that we can see in this instance that liver cancer would appear to be a product of phenotypes such as the cell adhesion, so the cellular matrix and how cells interact with each other, basic cell signaling phenomena, the ERB2 signaling pathway. But also of interest is we can see that it's also being driven by things such as how the immune system is responding. And again, all these are wildly significant associations of these networks. Okay? So, and then just to focus on more detail a little bit, so this is the proto-analysis for the immune response here, and this is meant just to show real data, so you don't have to completely trust me. We get a significant difference in the interactions in the antigen processing network, suggesting that how one's capacity to monitor their immune or maintain their immune response may be important in determining who is developing liver cancer. So what we've done is not only look at liver cancer as an outcome, but we've also looked at the phenotype of non-alcoholic fatty liver disease, shown here, which is a common cause of chronic liver disease, and commonly through non-alcoholic liver disease and non-alcoholic steatohepatitis actually progresses to cancer, but in and of itself can be a fatal disease. So what we've done is then use an alternative common data resource, a common disease data resource, liver disease, ranging from fatty liver all the way to steatohepatitis, to look to see if we can find common related mechanisms that would drive that process as well. So in doing these analyses, well, just quickly, my one obligatory gross sort of pathology kind of picture, shown here on the left is your liver, your normal liver, and on the right is your liver when you're obese. And in fact, just a 30-second digression, one of the fascinating things about the liver in its biology related to obesity is the liver is one of the primary locations where individuals store fat. And if you think biologically and evolutionarily, that makes a ton of sense, because the liver is also where you metabolize fat. So if you actually need ready, rapid stores of energy, you want to have the fat reposited where the machinery is available to take that apart. The evolutionarily interesting analog, though, is that clearly we were not well designed to deal with excess calories. And in fact, the liver has very poor mechanisms to quit storing fat. So what happens is, as one becomes increasingly obese, one has increasingly large fat stores in one liver to the point that it can no longer function, and it becomes fatty liver, as well as then results in morbidity associated with having so much fat that's interfering with how your liver can perform its normal functions. So we looked at, again, at a large-scale study with literally hundreds of thousands of variants, but in this instance now, looking at multiple diverse phenotypes associated with liver disease, fibrosis, steatosis, cirrhosis, hepatocyte ballooning, lobular inflammation, and how all these would come together, and then started to look, can we now take these millions of or hundreds of thousands of variants and look at how they might produce risk or change or be different among individuals with or without these phenotypes? And what we can see here is, interestingly, we see a variety of novel of pathways that emerge, but again, not completely surprising pathways. So we have the biosynthesis of steroids, cholesterol biosynthesis, protein translation. But interestingly, in this completely independent phenotype, we also see that we see antigen processing and presentation, suggesting that, in fact, there may be common underlying biologic processes that are driving one to not only have these particular phenotypes, cirrhosis, fibrosis, non-alcoholic steatohepatitis, but also that drive one to go on to and progress on to liver cancer. So lastly, our current focus is, with these proof of concepts, are now driving into assembling very large collections of publicly, at least published data sets that in theory should be publicly available. So if we just do our literature search and follow through with the paradigms of data sharing, in theory there are 54 type 1 diabetes data sets, 13 obesity data sets, 12 liver disease data sets, for a total 79 large-scale multi-SNP data sets that should be accessible. I will say, perhaps a little unfortunately, when we go to the public resources where these are supposed to be registered, we can see instead of there being 79, there are 9 available. But that said, we can see from these 9 available data resources that we can easily access, we can assemble now up to tens of thousands of individuals to start to dissect our individual phenotypes. So shown on this slide are the slides that are the studies that will allow us to look at obesity, diabetes, and begin to take apart, because what you'll see what one sees in the obesity studies, diabetes studies, is that they all have common underlying phenotypes. So our current work with our NGCC activity is now to do what we did in the more focused, limited data sets, take this very large omnibus data set and look at these collections of different sub-phenotypes that are part of the obesity phenotype, and performing the analysis to map the individual genes and the biologic processes that are underpinning these specific diseases to a common outcome, so that we can start to take and examine the emergent properties, how these networks drive these components, and then look at the common phenotypes, the outcomes such as diabetes, liver cancer, and heart disease, and see how those networks then come together through these individual subphenotypes in order to produce an emergent whole that's different than the specific sum of the individual parts. So in summary, we believe that common disease is a product of multiple genes and environmental factors. We believe that approaching common disease as a complex adaptive system will allow us to embrace its complexity and assist us in finding the emergent properties associated, the interaction and the places where the whole is more than the sum of the parts. We believe that we can do this effectively against these very large data sets by the use of a data science instrument that allows us to capture this data and manipulate it in novel high-performance ways, not unlike the way that Google and other groups are doing it. We are focusing originally on the emergent properties at the gene level of looking at how the networks themselves, again, a common analytic tool that Google, Facebook, Amazon use to find the interaction between components. And part of what we're interested in doing is then using these network models then to better understand how diabetes, heart disease, and cancer emerge from these underlying network signals. So with that, I'll close and go to take questions, but want to leave up, we're actively looking for groups and individuals that are interested in participating in collaborative activities. I will leave my email up for the remainder of the question and answer period here. And look forward to the questions and answers, but please feel free to email me if you would be interested in either leveraging or exploring more as to how you might use our next generation cyber capability as a data science instrument, as well as would be interested in collaborating in any of our obesity and cancer related studies. So with that, I'll take questions. I see we have a couple of questions before us. Let me start with the first question. So if you have questions, please feel free to enter them into the Q&A box, and I will then read those questions that come to us, and then use and answer those. So please feel free, if you have questions related to the presentation or other, please add them in. So the first question that I have is, would not the differences in the quality of the data sets limit the ability to work with these data sets, and how would one compensate? And I think that's unequivocally a key question associated with any of these big data sets. So part of what we do that I go through too quickly in order to cover as much ground as I did in this particular presentation is we do a lot of data quality assessment of the individual data sets as we receive for robustness and reproducibility. Certainly within the molecular data sets, there's a series of quality assessments we can do. And then also in the molecular phenotypes, they can be quite uneven. So part of what we do is certainly where we evaluate the metrics, we can enter weighting variables in where we know differences in quality, where we can differentially weight the contributions of those particular components when they're of different quality, and where we don't necessarily know these, we can actually then, through the multiple data sets, we can actually look, there's a whole variety of statistical methods that allow us to look for the influence of particular data or data sets, and we can make sure that certain data sets, even if we don't know in advance the quality, are not differentially influencing the outcome. So there's a variety of analytic attacks that we will take to attempt to compensate. So it's a very good question. Any other science? Do we have other science questions? Again, someone has asked, where can they contact me? I think you should be able to see my email address. If not, I'll say it out loud. So you can reach me at Kenneth Buetow, Kenneth Buetow, Buetow, at asu.edu. So that's my email address. I've also been asked whether one can actually access the materials from this presentation. I believe the whole presentation, both the tape form as well as the slideware, will be available from the conference site. So anybody who's interested, who may have come in late or would want to learn a little bit more, feel free to look at the presentation, go through it again, and or look through the slides. And we would welcome anyone, again, follow through with questions, and if you look there, my email will be embedded in that as well. I have another question, is how can we use big data into applications into industry? Well, one of the things I would share with you is that the infrastructure that we've created at ASU that has the aggregation of these types of data, we're interested in working with industrial and commercial partners and leveraging both the platform as well as collaborative research activities. So if people are interested, moreover, the infrastructure that we're creating, we realize not everyone is interested or capable of working with academic groups, is all open source software. So the software capabilities, so in particular, the Mahout framework and all those other are all the Apache Foundation supports and distributes the basic core foundations. All of the tools that we generate, so for instance, I mentioned the PODA analysis, the pathway of distinction analysis, that software would be available for anyone who wants to use it in any context without restriction. So do we have any other questions? I'll wait a moment or two more if people want to add. Another good question I got is, does our infrastructure support GPUs? One of the components that I didn't spend much time on is that the NGCC actually has, as part of its high-performance computing component, a collection of GPU processors. So if there's specific software that we need to run that does unique analysis, we can actually use GPUs. So that's part of the framework as well. And we can arguably and hopefully go back and forth between specific tools that would do unique things in a GPU environment and bring that back in through the data representations and data stores and use CPUs and GPUs simultaneously, or in concert and or in pipelines. I have another question that says, under trends and forecasts, how can these data sets help and what kinds of actions to implement in the future? Can we determine the cause from the effects, i.e. in type 2 diabetes? I think a little unequivocally that's a challenging question that one always has in approaching any of these problems. One of our strategies for looking at the difference between cause and effect is by using temporal data sets. So one of the data sets that we're hoping to be using in our analysis is a longitudinal data set. So where we have clinical records that occur over a 40-year period. So we can actually see progression over time in particular phenotypes. And if we then have other variables that either are invariant, so one of the reasons I mentioned, for instance, constitutional variation, is we can use, for instance, constitutional variation or germline variation as an invariant component. And we can then look at, through semantic variation, changes that are occurring over time. We can similarly do that with other types of measurements, but since we're in a genetics and genomics seminar series, I thought I'd focus on those to begin with. But there's other trends that we can use, or looking at the trends of the data when we have time variables that help us distinguish those as well. Other questions? There's been a question that was raised, Ken, about batch effects. And unequivocally, one of the challenges one faces even within a data set is the notion that different things can occur, or there are measurements that occur based on the timing of when one either does one's molecular analysis or when one has done the specific time scale associated with phenotypic measures and others. Our strategy, again, for that will be to be sensitive by doing divisions of the data sets, both by looking at the influence that comes from specific contributions when we're combining multiple data sets, so making sure that individual data sets do not have disproportionate contribution, or at least being aware of what the individual contributions are of specific data sets, but then also question that we have is, let's see, from my personal experience, can tell how will big data studies be useful in tackling with diabetes and other combinations? And I actually think big data is part of the essential piece of what we're going to need to deal with. And so again, while I very much understand and appreciate the importance of smaller scale directed experiments associated with testing specific sub-hypotheses and experimental models, I believe that we will only be able to reassemble the whole through big data approaches by looking at this continuum class of observations and the ability to synthesize and integrate those components. Now, many of my colleagues say that that's only hypothesis generating, of which I'm quite comfortable with. So if we only are drawing conclusions that still need to be tested in more controlled situations, outstanding. But at least we've made those connections, and we don't have to test all possible combinations of activities. So I think it's already demonstrably useful in looking at the analysis and reanalysis of large-scale data sets, as well as having the ability to do and identify emergent properties. I'm looking to see, are there any other questions? It looks to me right now that I've addressed all the questions. Again, I'll remind people that the talk, if people are interested in accessing the talk, both in terms of its presentation as well as the slideware, that'll be available through the conference site, and I'd encourage you all to get that. Those of you who are interested in learning more, please feel free to contact me. As an academic group, we're always interested in collaboration and interaction, so anything that we can do to help with that, we would love to actually do that. My email is is listed on the slideware if you want to come back to it later. And again, you can reach me at kenneth.buetow, KENNETH.BUETOW @asu.edu. So let's see. So I have one more question here. What's my take on personal genomics companies? Actually, I think personal genomics companies, if the work is done with high quality and with rigor, I think actually provide very important information for people to be using in their personal decision making, as well as in their portfolio of further participation in the biomedical enterprise. Any other questions? Well, I want to thank everyone for their time and attention. I always thank you for the collection of questions. Please feel free to let me know if you're interested in more follow-up, or if I can help with clarifying other materials. Please feel free to access the materials that are part of the conference on the website. And with that, I will say thank you and let you go on to your next session. So thank you very much for participating and interacting. Bye-bye.